Hi guys, welcome to Pathopulse_Radhika. So today we are going to discuss about an important topic and the very most basic topic about pathology. It's about the cell. So we have a human body. The human body is made up of so many organs. So how is these organs made up of? Okay, so you have various organs. So you have a smallest structural and basic unit of life which is called the cell. So many cells. So this is how a cell appears. So you have a nucleus, you have a cytoplasm. The cells will make up tissue and the tissue will make up an organ. That's how a human body is being formed, right? So when you take the cell, the structure of a cell, a diagrammatic representation, this is how a cell appears. So you have a cell membrane, then you have cytoplasm, a jelly-like substance in which your various organelles are floating, your nucleus, your nucleolus, your endoplasmic reticulum, mitochondria, and so on. So this is just a 3D view of how a cell appears. So you have a cell membrane, the plasma membrane, which is having a protein scaffold, and they have something called cytoskeleton. Okay, the framework over which the cell rests. Okay, so you have a cytoskeletal framework like a scaffold. Then inside that you have cytoplasm, which is a jelly-like substance. So inside this cytoplasm there will be so many organelles which are floating, like your endoplasmic reticulum, mitochondria, your ribosome, centriole, and so many organelles. And let's see about each one of them. The first is endoplasmic reticulum. So if you take endoplasmic reticulum, they are present near the nucleus and they are these folded structures. And you have the smooth and the rough endoplasmic reticulum. It's called rough because over the surface you have got these structures called ribosomes, which makes them appear rough. That's why they are called rough endoplasmic reticulum. Whereas your smooth endoplasmic reticulum doesn't have ribosome in them. The main function of endoplasmic reticulum is transport of proteins. Whereas in addition to that, the rough endoplasmic reticulum, because they have ribosomes, they synthesize proteins. So these ribosomes helps in synthesis of proteins, and your endoplasmic reticulum helps in transport. Reticulum means network, like they appear like a network. Okay, that's why they're called endoplasmic reticulum. What's the function of smooth endoplasmic reticulum? They are involved in synthesis of steroid hormones and lipoprotein synthesis. Okay, that's about your endoplasmic reticulum. Then comes your mitochondria, the powerhouse of the cell. As you all know, it is involved in generation of ATP, the energy booster of the cell. So ATP provides energy for the various cellular functions which happens inside your cell. So it's called the powerhouse of the cell. The next is the nucleus. So the nucleus has got the DNA, the genetic material. Okay. Then comes your lysosome, which helps in waste disposal. So they have a lot of enzymes like hydrolase, protease, and various other enzymes which helps in digestion of these structures. And these lysosomes have an acidic pH. Okay. So these are about the various organelles, and you have something called Golgi body. So your endoplasmic reticulum, your protein is synthesized, they are transported, and some sort of protein modification or protein assembly happens in your Golgi bodies. Then you have these structures, barrel-shaped structures called centrioles, which can produce spindle fibers, which helps in cell division. This is about the various organelles. Okay, and you have two other organelles which many of you tend to get confused. One is proteasome and the other one is peroxisome. Proteasome, as the name indicates, proteo means protein, it's involved in degradation of proteins, whereas peroxisome is involved in breakdown of free fatty acids or fatty acids, and then they will scavenge your free radicals. They will detoxify your free radicals. This is about the various organelles in the cell. Now a quick recap. You have your cell membrane with their cytoskeleton, and the cytoskeleton is made up of proteins. You can remember it as AIM. The mnemonic is AIM. A for actin microfilaments, I for intermediate filaments, and M for microtubules. So your cytoskeleton is made up of these proteins. Then comes your nucleus with your genetic material, your nucleolus, which helps in synthesis and assembly of your ribosomes. Okay. Then comes your endoplasmic reticulum. Your endoplasmic reticulum is involved in transport of proteins. Rough endoplasmic reticulum, they have ribosomes which can help in protein synthesis. Your smooth endoplasmic reticulum is involved in synthesis of steroid hormones and lipoproteins. Then your Golgi apparatus, which helps in protein modification. Your lysosome helps in waste disposal, and the mitochondria, the powerhouse of the cell. So this is about the various organelles inside your cell. Okay. Now we saw a diagrammatic representation. Now you must know how a cell appears on a H&E stain. So what is H&E stain? So let's start from the beginning. Imagine a person has undergone hysterectomy. The uterus is removed from the patient and it goes to the pathology department. You can't visualize the entire uterus under the microscope as such. So what we do, we put it in formalin, we cut it, we fix it, and we process it. So there is a whole lot of procedure which goes in the lab for 1.5 to 2 days. So I'll just tell you what's important for this particular presentation. So we cut them into small pieces and then we process it, and then we take certain sections using a blade and then we put it over a slide and then visualize under the microscope. So for seeing this, you can't see as such. You have to add certain colors so that you can see the nucleus and the cytoplasm beautifully and clearly. And what are those colors or what are those dyes? So those, one of the most common thing used is hematoxylin and eosin stain. Okay, so why do we use two colors? Hematoxylin will appear blue on staining, eosin appears pink on staining. So certain structures appears blue, certain structures appear pink depending upon the chemical property that we'll discuss in another video. So as of now, remember to visualize this as we use a H&E stain. The nucleus appears blue, the cytoplasm appears pink. So this is one cell, this is another cell, this is another cell. The nucleus appears blue, the cytoplasm appears pink. So this is how a cell appears on a H&E. Why am I showing this is because in the upcoming videos we'll be showing so many histopathology image. So you should know what we are talking about. So you should understand the basic how a cell appears, then only you will understand the other stuff, right? So this is a cell, this is the nucleus, this is the cytoplasm. Whereas if you see this one, you can see some small dot-like structures inside. So those are nucleolus. Okay. So this is how a cell appears on a H&E stain. Right. So now we have seen about the diagrammatic representation of a cell, the various organelles, and how it appears on H&E. Now let's take a small quiz about the various organelles. So you can just proceed along with me, or you can take a paper and write down about the organelle. So number 1 is your nucleolus. What is number 2? Very good, it's nucleus. So what is 3? Network-like structure which is surrounding the nucleus. It's a rough endoplasmic reticulum because they have ribosome. Next, coming to 4. I have not taught you about 4. 4 is nothing but small vacuoles which store certain substances, right? Next, what is 5? 5 is nothing but these ribosome, these small blue round structures. Where is 6? What is this one? This is also folded structure, but it's a little away from your nucleus, so it's a Golgi apparatus. What about 7? So 7 is in the outer side like a fiber. So what is this? It's a microtubule which is in the cytoskeleton of the cell. Next, coming to 8, what is this? This is also a network-like structure, but it doesn't have ribosome. It appears smooth. So it's a smooth endoplasmic reticulum. And what is the structure 9? It has got cristae. So it's a mitochondria. What about 10? 10 is lyso. It's a vacuole. It's again a vacuole like your 4. Next, where is 11? So 11 is that in substance in which all these organelles are present. So it's a cytoplasm. Where is 12? So 12 is They have represented all diagrammatically same, but it's actually a lysosome. The waste disposal, helps in waste disposal. So this is about the various organelles, and we have 13. Number 13 here, what is this? The barrel shaped structure, the centriole, right? So this is about the various organelles inside the cell. So I hope you're clear about the structure of a cell and how it appears on histology.